Good morning, everyone, and welcome to the next session in the series, What About Privacy and Progress in Whole Genome Sequencing? My name is James O'Leary. I'll be moderating today's discussion. As a reminder, this session is part of a longer series, the title being Technology is a Deeper Dive Needed. We're joined by a number of great speakers today who will go through their presentations, and then we'll have questions at the end of the webinar. As a reminder, everyone is muted on the phone, so please type in your questions using the chat box on the right-hand side of your screen throughout the webinar. Send them to us as you have them, and we'll compile them on our end and get them ready for the speakers. In addition, these sessions are recorded, so if you'd like to listen to this session later or even send it to other people to listen to, they should be up within a week. You can also visit our website at geneticalliance.org/webinars to listen to archived versions of the first 8 webinars. We will have 2 more sessions in this series following this one, and you can also register for those sessions online. So without further ado, we'll get started. Our first presenter is Yaniv Erlich, who is the Principal Investigator and Whitehead Fellow at the Whitehead Institute for Biomedical Research. Yaniv will introduce this topic, Technology is a Deeper Dive Needed. Welcome, Yaniv, and we'll switch to your slides now. Okay, we can view your slides. Thank you.
Terrific. Thank you very much for, it's a pleasure to be again in the Genetic Alliance webinar. And I'm going to talk today about new techniques to breach the privacy of whole genome sequencing data, what I would refer to it as genome hacking. Now, I would like to start my presentation by stressing the point that public data sharing is highly important for genetic studies. We cannot move forward and fully realize the value of whole genome sequencing or in general of human genetics without sharing the raw material between researchers. And just in my own group, examples for that, where we identified the 2 genes for devastating pediatric disorders. And we could not do this process without getting access to the 1,000 genomes data sets, to DBSNP, and other wonderful resources that are out there. So basically, we couldn't give hope to these patients and their families and their parents without public access to genetic data sets. Now, in our view is that in order for this endeavor to be sustainable, we need to map the risks for privacy and to consent participants about these risks. And this was the rationale for the following project. So about a decade ago, or a bit more than a decade, it has been realized that there is co-segregation between the Y chromosome and surnames. Let me explain that. So for instance, here we have the Smith couple, and if they have a child, then the father will give his child the Y chromosome, if they have a son, right? And in most Western societies, also his surname. Now, if this son is getting married and also has a son, he will give him his Y chromosome and also his surname. And this process will continue and will only break up by non-paternity events, explicit surname conversions, and by mutations that will drift the Y chromosome. Now, the recreational genetic genealogy industry has took advantage over this correlation, and now they offer services that they will send you a cheek swab, a swab to sample the DNA on your cheek, and then you can basically put it in an envelope with $200. This is very important, and then they will genotype a series of markers on your Y chromosome that are called short tandem repeats, STRs. And here you see an example, and then they will upload this information to a website. For instance, ysearch.org, this is what you see on your screen right now. And so you can upload this information with all these markers on your Y chromosome and also your surname. And in fact, what I show you over here are my own test results. And the reason that people are interested in these tests is because it's a lot of fun. You can go and you can find a match to your patrilineal relatives. You can reconnect with them, find a black sheep in your family, and so on. So this is a lot of fun. And what we thought was maybe we can exploit these resources to identify whole genome sequencing datasets. So let me show you how it works for Craig Venter. So we took the whole genome sequencing data from Craig Venter from the NCBI. We just downloaded the data from the website. And then we extracted the short tandem repeats on his Y chromosome using Lobster. This is a tool that we developed in the lab about a year and a half ago. It was published in Genome Research. It took us about 30 minutes to go over his genome and identify these short tandem repeats. So here is an example for a marker on his Y chromosome called DYS458. We found that there are 17 repeats for this marker. So then we went to ysearch.org, a public website that you can search in these recreational genealogy databases, and we just inserted 17 that this locus, this STR, has 17 repeats. And we inserted all the other markers that we could find on his genome. And then we clicked search. And after a few seconds, what we got was that Venter, the surname Venter, was the top match by searching this database. So you can also try it yourself, because this is an internet study. So if you would go to bit.ly Craig_Venter_haplotype_updated, if you just go to this link, it will redirect you to ysearch.org, to this web page with all the markers of Venter from Craig Venter inserted in the web page. Just click search and see that you're going to get Venter as your top match. But basically what I just showed you is how we can take whole genome sequencing data of a person, of a male, and using this process and public internet resources, we can infer the surname of the person. Now, we got Venter, we got the surname Venter, but can we really get to the person, Craig Venter, from this data? So here is what we tried. We know, based on the genomics data, that the surname of this genome is Venter, and that he is a male. Now, in many genomic studies, we also know the state and the year of birth. And these are not considered as identifiers by the HIPAA safe harbor privacy rule. So you find them in some studies in genetics. So then we just took this combination of identifiers, Venter, male, California, someone that was born in 1946, and we went to ussearch.com and peoplefinders.com. These are engines to search in public records using these identifiers. So we just searched, and we got, for this profile, we got 2 matches, including our friend Craig Venter. So again, we can take whole genome sequencing data, and just using internet tools and some minimal information that is posted online in the study, we can get very close to the person in some of the cases. But of course, the question is, with Craig Venter, it's a bit of a cheating, right? Because we knew that this genome belongs to Craig Venter. The main question is, can we identify anonymous personal genomes? And this is what we try to do. We focused on the 1,000 Genomes project on 10 genomes that were collected part of the Utah population. Now, it's important to note that the genomes in the 1,000 Genomes project were consented that there might be, their consent was done in the beginning of the millennia, but the consent form stated that there might be a breach of privacy in the future. And this what gave us the green light that we could actually conduct this study and test if we can identify these genomes. So we took 10 genomes from the 1,000 genomes that had good coverage, and we used Lobster to get their YSTR. And then we just went to two databases, ysearch.org and another database called smgf.org, and we searched the YSTR profiles in these databases. And for 8 of the genomes, we could get certain predictions of people with Utah ancestry, which is a good indicator that what we are getting might be true, the true surname. But let's test that, right? Okay, so we decided first to focus on this pedigree that you see over here. This is a 3-generation pedigree. All the individuals in this pedigree donated their DNA to genetic research. You can get the DNA in the Curiel database, in the Curiel cell repository. We were able to infer the surname of the 2 grandfathers that are labeled in black over here. So we got the 2 surnames, or 2 surname predictions, and then we did something very similar to what I show you over here. We don't disclose the exact details to respect the privacy of these families. But we just, in Google, we just put the surname and a few more identifiers and click search. And the top match, the first link in Google, was an obituary of a family in Utah with the exact description of the pedigree that we have. What do I mean by that? All the details were matched to the pedigree in the Curiel database. First, the family was from Utah. Second, the number of kids in the obituary matched exactly to the number of kids in the pedigree. The birth order, male and female, matched exactly to the birth order of the kids in the pedigree. And think about it, every time, every kid is another 50% chance. If it's just a random chance, there is a 50% chance of getting it wrong. But it was exactly the same. The surname of the paternal grandfather matched in the obituary matched to the one that we inferred from our prediction. The maiden name of the mother matched to the surname that we predicted for her father. And what are the chances? Both of them were surnames. And also we were able to test the age of some of the individuals in the pedigree, and it matched what we thought, what we found for the people in the obituary. So then we calculated what is the chance that this was just, random match. And the chance after scanning all households in the United States was 5 times 10 to minus 9. Terrific. So there is a very low chance that this was just a random match. But then we thought, you know what? Maybe this was just beginner's luck, right? Okay, we could do that for one pedigree. But in fact, using the same or very similar process, we could also get another pedigree from the 1,000 genomes and another pedigree. Every individual in black over here is someone that we could successfully infer his surname. And the probabilities that these were just random matches were very low in all cases. In fact, we have so much data at that point that we could trace the genealogical relationships between the people that are part of the 1,000 genomes and the people that donated their data to the recreational genetic genealogy study. For instance, in the second pedigree, you see here the grandfather of the father of the mother. His second cousin once removed, donated his DNA, and by that identify his entire family. And we have other examples that we don't, the take-home message is that we don't need the same person to be in the recreational genetic genealogy database because of the correlation of surnames in the Y chromosome. This correlation can go several generations ago. So your second cousin, third cousin, and so on can identify you if they participate in one of these activities. Now, after we did this process, we could identify all the people that donated their DNA to these studies, all the people in black, which was close to 50 CU, 50 samples, part of this Utah collection. So I showed you an approach how to breach the privacy of research participants in some cases that doesn't involve any experimental work. You just need a computer and internet access. The information propagates via deep genealogical ties. So it means that even if some patrilineal relative that you don't know participated in this recreational activity, he might identify you. And everything here was done using public resources. No access, I didn't need to hack to any computer or to break any password. Everything was done using stuff that appears online. And I just go quickly on the recommendations that we have. So we think that part of the consent of participants, we need to be honest about the risks. We need to tell them that there is a risk that someone might be able to identify them. Of course, we should also be very honest about the benefits, and there are terrific benefits for genetic studies for society, and we should tell them about the benefits and let them decide and respect their decision. Maybe we should adopt some multi-tier approach where we can give participants options for data sharing. Some participants maybe would like to shy away and not share their data. We should respect that. Some of them will be more active and more and would like to disseminate their data, and we should also enable that option. We also recommend to maintain these proactive approaches to map the risks. I think that when me or my colleagues do something like that, is what's called friendly hacking, this is far better than that a real one would happen, right? This would really destroy trust in genetic research. And we should also think about some technical solutions, because it seems that we didn't explore enough these solutions, and should think how we can go forward. So I would want to just to acknowledge a terrific group of people, really. I was thrilled to work with this group of people, especially Melissa Jimrick. She's a Harvard MIT PhD student, and she did most of the work that I presented. Our paper was published a few months ago, and it's available with registration to Science. And thank you very much for listening.
Great, thank you so much, Yaniv. I think you did an excellent lead-in to our next speaker, who is Robert Shelton, CEO of Private Access, where, well, Yaniv talked a lot about how technology can be used to breach privacy. Robert will take the other angle and talk a little bit about technology and solutions that can help protect privacy. We'll switch the slides over to Robert now. And Robert, you should be able to select on your screen to take control.
Off mute, and I think I've just achieved coming off mute. Yaniv, thank you for the setup. During your remarks, I decided that maybe Eric Schmidt was correct in his statement that the only way to protect privacy is going to be for youth to change their names at the age of 25, and that will be their best shot at privacy on the internet. And that's a little bit tongue-in-cheek, and I know that Eric took some goading because of that comment, but what we focused on at Private Access is looking at the aspect of privacy as trying to strike a balance that each individual tries to do between the aspects of having privacy and having the access to information, where the recognition is that most people have different views about privacy. This document that I'm showing reflects the results of a 2009 study that was commissioned by the Institute of Medicine by Dr. Alan Weston. And what Dr. Weston tried to ascertain was what were different perspectives of the interviewed parties regarding how open were they with their data and how much information would they be willing to have available and what would they require before their information was made available for research purposes. And as you can see, what we got was a skew between people that didn't want researchers to contact them or to use their data under any circumstance, roughly 16.5%, and then the other extreme at 1.5% that said it was okay for a researcher to use their data without their consent for any reason whatsoever. But between those two spectrums, roughly 85% of the remaining people said that they wanted some opportunity to express whether or not their data could be used for their purposes that they were interested in having and precluded from use for reasons that they did not want. And so what we've focused on at Private Access is the idea that in order to allow consumers to actually get in the game and for the great abundance of that bell curve in between those two extremes to get in the game, there needs to be a fast, simple, and intuitive way for consumers to express their view, and that needs to occur in real time, and it needs to be brought down to very simple terms of permitting, i.e. consenting, declining, or asking time to learn more information or to get back to the answer of whether they permit or decline to permit at a later date. We have brought those notions of that simplicity of either allow, deny, or requesting the opportunity to ask me later when there's more information available about the specific reasons for the proposed use of the data, down to a series of granular settings that in this user interface for a system called Trials Finder, we broke out as being controls over what advocacy and support groups could use the data for, medical researchers and what they could use the access to the information for, and then various data analysis platforms. And we've broken it into three different categories: a category of permitting the de-identified data to be discovered, permitting contact information to be connected with that de-identified data, and then permitting the use of the data, meaning for it to leave the registry and to actually be moved over to the control of another party, as being the opportunities for those granular consents. And our notion here was that that privacy technology, that a technology for reducing the wishes of the consumer as relates to their information was absolutely necessary in order to realize the social goals of getting the access to information that most patients want when they need it, and the privacy that many people want when the access is less necessary. There's generally been an attempt to, or a slippery slide in terms of conflating the notions of data security, which means things like encryption and authentication and identity verification, with the notion of privacy. And what we've tried to do is to focus on that fourth aspect, with not denying the need for the other three. The other three are absolutely necessary, but they are not sufficient in terms of getting at the notion of access controls and consent. They are just precursors to obtaining privacy. And so what we've tried to focus on is that last fourth element. Now, recognizing that consumers are most likely going to feel like they've been thrown into a category of not knowing how to set privacy settings in a really useful manner, we've also developed a meta language around the notion of establishing guidance from various leaders or decision influencers that might have opinions to express with respect to the topic of how much data they would suggest be shared, for what purposes, under what circumstances, that would allow each consumer, as opposed to coming in and starting from a blank slate, to make a decision either, if they wish, to start from a blank slate and set their preferences manually, if they didn't want any influence, or to turn to someone in their network or in the larger society that they found having credibility in terms of assisting them, and to look at what those leaders, those thought leaders suggested that they might consider, and then use that as a starting point for setting their privacy preferences. We have put this in use inside of a platform that Private Access is building in concert with the Genetic Alliance called the Pier Platform, which stands for Platform for Empowering Everyone Responsibly for Medical Research. It is a cross-disease, cross-organization resource as a registry that has a unique governance structure, and that governance structure is, it's up to each individual to set their privacy, determine their privacy wishes through the technology I just summarized for who they want to see their information, under what circumstances, and for those decisions to be set out in a easy to use, patient-centric, and dynamic form through the technology that we call privacy layer. On each side of privacy layer, we have data entry facilities on one side for consumers coming in, setting their privacy preferences, but also making accessible their health data and their contact information through core components, but then those components being capable of customization on the outside of the core of the platform. And then on the other side, for data query facilities, with again those core capabilities of data query, focusing on just the permissible information, which means the health data and the contact information that is as mediated by the privacy directives from time to time through a portal that could be customized. And you see some of the brand names that we have actually built out customizations called Recruit Source, which is recruitment for clinical trials on one side in orange, and Trials Finder, Reg for All, and a program called Free the Data, which is focused on the BRCA gene mutations on the other. These capabilities we think are useful to lots of people, and so what we've done with Pier is actually to establish an ability for the platform to be iframed, meaning to be dropped into, in a technological, a web-based technology, to be dropped into any website, so that any website, in this case this is a rally for liver disease awareness that's occurring in a couple of weeks in Washington, and for them to start collecting data and giving individuals the right to control privacy over their information would be virtually impossible, except that through the ability for iframing in the platform of Pier, we're able to drop in to their website the entire Pier platform, so it appears in a customized form with their look and feel of their website and with all of the capabilities of the Pier platform made available to them, so long as they are willing, as the organizer, to give the individuals who contribute the data the right to set their privacy preferences in accordance with their wishes. And so this area that's highlighted reflects the outlines of the widget that comes in through the iframe to any web page, and then it's able to be customized with the logo and the, of the organizer, the call to action that they wish to set, and for all the fonts and background colors and links to follow the theme of the website so that it gets to be somewhat transparent to the user that this is going on. By the same token, because most sites don't contemplate this kind of data sharing and the privacy permissions being delegated back to the individuals, the peer network's privacy policy in terms of use are accessible, as well as earned certificates, merit-earned certificates for the trust associated with those. And then, as an added value, each of those platforms is able to establish badges which can be easily uploaded in a few lines of code to other websites in their consortium. So in this case, the rally organizers are able to give the various sponsors, partners, members, disease advocacy organizations a badge that can go on their web page, where they are able, anyone visiting their web page can start at that page they're already on and get started with setting their privacy preferences, entering their health information, and making their data accessible as and when they wish. So that's just a glimpse of some of the notions of privacy from the perspective of private access and our work with Genetic Alliance. And I'm going to turn this back to James at this time, and for Debra to continue.
Great. Thank you so much, Robert. So now I will turn things over to Debra Mathews, Assistant Director for Science Programs at Berman Institute of Bioethics and Assistant Professor of Pediatrics at Johns Hopkins University, who will react to the first two presentations and talk about some of the deeper issues of technology and privacy and whole genome sequencing. Thank you very much for joining us, Debra.
Yes, and thank you very much for having me. Can you hear me okay?
Yes, I can hear you, and we're just waiting to see your slides.
Oh, I thought they were up. Are they not? Can you not see them?
Not yet. There should be a button on your screen that says "Take control of the presentation."
Okay.
Or show my screen, it says, I believe.
I thought I had clicked it. Oh, there we go. Okay.
Okay,
now
great.
Can you see me?
Yes, we can. Absolutely.
Great. So Yaniv and Robert actually made my job incredibly easy. And all I'm going to do is I'll respond a bit to what they said and provide a little bit of an ethics frame for thinking about how these changes in genetics and genomics impact people who are participating in research. So I want to sort of set the stage. Again, part of this is going to be recap. 4 forces in genomics and genetics that we're facing right now, or that are coming together to create a set of issues around privacy. So the first is the culture of openness in genetics and genomics. Of course, this has been the case since the Human Genome Project and the Bermuda Principles that said that everyone who was involved in the Human Genome Project was to post their data publicly within 24 hours, because, of course, as already has been pointed out, it's critically important that we share these data, because I cannot tell you anything about the meaning of your individual genome without first studying lots and lots of genomes and information about, information about the people from whom those genomes came. So I need lots and lots of data on lots and lots of people to be able to imbue with meaning your individual genome. So this openness and data sharing is critically important to the science. The second is the cost of whole genome sequencing, as of course was pointed out in the report and has been pointed out by many people. You've probably seen this graph, demonstrating the incredible pace at which the cost of whole genome sequencing has decreased. Of course, the first genome cost about $2.7 billion to sequence. That was less than 15 years ago, and now we're looking at a bulk genome price around $5,000. And it's anticipated that this price will go down. And of course, with a decreasing cost comes increasing utilization. So whole genome sequencing is increasingly being incorporated not only into clinical care, but also into research and into direct-to-consumer products of the sort that Yaniv talked about earlier. The third force is the advent of social media and networks. Although they haven't been around that long yet, they've all been completely integrated into many of our lives. And one of the things that these sorts of social media, social networks have provided is a sense of control over our information. Some of that may be illusory, but within Facebook, I can decide for any given picture or post who I want to see that information. I can say just my friends, just my family, just one particular network, or the whole world. And many people have come to expect that we will, in fact, have some control over our online personas and our online information. And this doesn't happen exclusively in the context of, what you had for breakfast and what you did on the weekend, but also our health information. And there are many websites now, including those that Yaniv and Robert talked about, where you can upload your data and learn from it, explore it, share it, and use it for many, many different purposes, including, for example, conducting your own clinical trials on your data that you have freely shared. And that's sort of turning on its head the idea of who is the researcher and who is the researched. And all of these things are changing sort of the relationships between researchers and research participants or research subjects in a way that we didn't really imagine previously. And of course, here's Yannis' paper, and this was not the first paper that questioned identifiability or the notion of identifiability within genomic research. It started in 2008 with a paper that demonstrated that we could in fact identify the presence of an individual in a pooled DNA sample. And that was sort of, if that was the first shoe, Yanni's paper was the second shoe to drop. That, again, threw into question the use of identifiability as a marker or as a decision point when it comes to human subjects research. So as you may know, whether or not something constitutes human subjects research and therefore comes under the umbrella of all of the policies we have that govern human subjects research can hinge on whether or not we consider something identifiable. And Yanni's paper and others have demonstrated that de-identified data are not necessarily as de-identified as we have thought they were. And the government actually acknowledged this a couple of years ago when they issued their ANPRM, Advanced Notice of Proposed Rulemaking, wherein they had 2 questions that asked whether or not we ought to consider human samples, tissue samples, and human genome data as identifiable in and of themselves. So this is an active question right now. So again, these 4 forces: culture of openness, the decreasing cost of whole genome sequencing, the advent of social media, social networks that are sort of pushing an evolution of notions of privacy and reflecting an evolution in our notions of privacy, our increasing control over our data, and the sort of decreasing strength of de-identifying sequence or samples. So when we think about these issues of de-identification and what that means for human subjects research, privacy, as was invoked in the report, is often what comes up. Privacy, although it means many, many different things to many, many different people, can be thought of as a condition or state in which cognitive access to personal information is restricted. Why do we care about genetic privacy? Well, it's most often paired with discrimination. This information can be used for both good purposes and for bad purposes. It can affect your social and economic well-being, your psychological interests, your relational interests. We often control the information that we share with different people, as I do on my Facebook page, based on the kinds of relationships we have with them, and of course, autonomy interests. And given all of these forces and our concerns about privacy, there are a number of different ways that we can try to approach the problem of privacy risks in genetics research, and a number of these were raised in the commission report. We can increase training. So of course, we should make sure that people who are touching the data appreciate that those data are in fact connected to individual people, and make sure that they behave appropriately and ethically. We can improve informed consent, as Yaniv mentioned, making sure people understand what the risks actually are and of course what the benefits actually are. We could scrap identifiability as a determinant of consent or of the invocation of our human subjects protections infrastructure. We can make clear that privacy can't be promised. And then we can sort of beef up our governance of the use of data and penalize data misuse. And all of these things, as a phrase that Robert used, are necessary but not sufficient. And this takes me to what actually both Yaniv and mentioned and Robert is doing, which is to provide some control. We could provide some control to individuals who are participating in research, and shift to these more participatory models that allow ongoing engagement, engagement of and control by research participants. And again, some of these arguments have already been made by Yaniv and Robert. So why control? So informed consent was originally devised as a way to protect or foster autonomy. And it was a fantastic way to do that. And it acknowledged sort of the limits of what we could do at the time in terms of communication, et cetera. But we now have the tools, as Robert very clearly showed you, that are necessary to develop more participatory models. We can, in fact, cede some control to the participants in the research. As I mentioned before, the norms of privacy have shifted, as have our expectations of control. So it used to be we wouldn't even talk about a cancer diagnosis. And now we wear pink and we run races, and the NFL is talking about breast cancer, and it's just a completely different world with regard to being open and forthright about conditions we're dealing with, our families are dealing with, et cetera. Also, the research community needs to earn the trust of the public. If we need, as we do, many, 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 many people to be willing to contribute samples and data to this research, they need to be able to trust that we're going to do the right thing. And it may be the case, as we talked about, as Yaniv and Robert talked about, that we should make clear what the benefits are. And there are many, many benefits of this research to society, but providing some control, providing agency in the context of this research might actually provide individual benefits that offset the risks of the privacy risks that are now very clearly entailed in this research. And I and others would say it's the right thing to do. And that's it. That's all I have. Thank you so very much.
Great. Thank you so much, Debra. So with that, we will turn things over to the question portion of our presentation. I just want to remind everyone that you can type in questions using the chat box at the right-hand side of your screen, and then we will get them and ask them of the presenters. If there are any questions that we don't get to, we can certainly send those to the presenters directly as well. While people are typing in questions, I just wanted to open it up to our speakers to see if you had any questions for each other.
Yes, so this is Yaniv and Robert and Debra. Thanks for wonderful presentations. I have a question actually for both of you. You both suggested or alluded to this notion of control of the participants over the usage in their data. Now, one question that I have, if you thought about the oversight that should be maintained. So, okay, so the participants can still, let researchers to use their data, but how do they know that they don't abuse the data or exploit it in some way? So if there is any how we can maintain this oversight?
So I can answer that as a practical matter. Debra, you want to answer it as a general principle, or do you want me to go first?
Oh.
Well, I'll interpret silence as I'm gonna go ahead and proceed and give you an answer from what we have done, Yaniv, in terms of the Pier Platform. We've actually, I think that we think of what we're doing as being more about trust than about privacy. So the issue from our perspective boils down to, do you trust the party that you are giving information to or permitting to use your information to honor their commitments as to how that information will be employed by them, and making certain that the terms under which you are making that access available are clear, concise, and unambiguous. And then, in addition to the controls for setting, I consent, I decline, or ask me later, those three positions, what we have is an audit trail on how the data was used at the other end by the recipient of that information, and an opportunity for a feedback loop to say that the consent was misused. And so it's not perfect. It only extends as far as the edges of the network that we've built thus far, but it's not difficult to imagine that if the right architecture and infrastructure were established, that those edges could be extended to first collaborative organizations, and then to sort of move to a trusted network that included organizations that were in collaboration. And yes, you could still have people that are screen scraping information by taking a photograph of it, or by jotting it down off of their computer screen, as opposed to permitting their behavior to be audited as they move through and apply the information. But our notion was that what we're fundamentally dealing with is trust, and if we can build the correct feedback loop, then the ability of the knowledge of people that they can lose a trust score would be self-disciplining over time as they would try to portray themselves in a desire to get more information as being high trust score recipients.
Great, thank you, Robert. Debra, did you have any thoughts on that?
Yes, sure. I think it's a critically important question, and one of the reasons, and I like Robert's answer very much, that it's about trust. I mean, in some research that we have done, focus group research with individuals regarding the use of their tissue samples in induced pluripotent stem cell research, we found that they would like to put limits on the use of that in particular ways that we might not as researchers anticipate. So for example, getting directly to your question about use or misuse, there were a number of folks who said, I am happy for pretty much anyone, any legitimate academic researcher in the US to use my tissues and data, but I don't want them to go, my tissues or data, to go outside the US, because I know what our protections are here in the US, and I can't guarantee that our protections will be enforced overseas. So I want to put sort of a geographic limit on the use of my data. And I think that then goes back, if you're giving that control to individuals, they get to make their own judgments based on their values about who they trust and who is trustworthy.
Great, thank you, Debra. And we did have a response from one of our listeners to this question. They said, perhaps oversight can come from institutional IRBs. Researchers should be obligated to apply to IRB for use of whole genome sequencing data from de-identified samples. Researchers will have to describe what they will do with the data, and hopefully IRB will decline any samples that they think do not work with the initial consent of people who donated the samples. Any thoughts or response to that?
So I'm part of.
This is Debra. I think another issue that. Oh.
Go ahead. Yeah.
Okay. So one distinction that's important, I think, is sort of getting consent prospectively and assessing what you can do with data and samples that were received under old consents, which are sort of two pretty different questions. And one is, how should your consent form look going forward? And the second is, how do we interpret old consents while we're thinking about this new research that we want to do today?
And I guess I would build on that and say that part of the enthusiasm that we have for the notion of the consent being a dynamic quality, more like an access control than a traditional consent. So if we think of consent as being opt-in, opt-out, if the ability is created to opt maybe, meaning opt-in, opt-out, between those two is I don't know yet, then the dynamic control can be put in place so that as technology changes, as needs for or opportunities for the usefulness of the data migrates over time, the limits established initially for the protections of the parties can be sort of seamlessly moved with additional information as it becomes available, and then a further access control permission or declining to make that permission.
Okay. Okay, so we are running a bit short on time, so I will ask a couple questions and ask that everyone give quick responses. One question is, are there any toolkits or websites that have been developed to educate patients about privacy and genetic information? Perhaps something that would guide patients through a series of questions to help them understand the potential risks and to consider their personal risk tolerance.
Well, I think I already answered that in terms of the peer network. The way that we've tried to do that is through the guide structure that we've developed, where we've attempted to get a cross-section of perspectives expressed so that each guide is actually asked not only to pick one position on the spectrum, but to answer as though they were approached by someone who said, let's say hypothetically to George Church, who we know is the Personal Genome Program, if you were very concerned about privacy, how would you set your wishes? And said to sort of the opposite party, who's a very rigorous privacy advocate, if you were unconcerned about your privacy, how would you set your privacy preferences? And so what we're trying to do is to get at that education program in a manner that speaks to the novelty of different conditions and circumstances, as well as parties' perspectives.
Great, and I will note from the Genetic Alliance perspective that we are continually building out information about this topic on our website, Genes in Life. That's GENESINLIFE.org, which is a educational website about genetics and health. Any other thoughts from our other speakers? Okay, great. So moving on, hopefully this is a quick question. Is private access able to connect health data stored in electronic health records with the patients who offer their consent via the PA system? Or does private access only involve the information that patients contribute on their own?
At this point, it's just patient contributed, although we have the architecture mapped out, and we are in the process of submitting a variety of grant proposals for building out the infrastructure. We think that the technologies are there to extend that to the EHR and beyond. So to the labs, to the laboratory data, to the X-ray data, to the pharmacy data, and to the clinical data, as well as the genetic information. So we're moving in that direction, and as aggressively as funding and technology standards will permit.
Okay, great. And so one last question, and if you want to wrap in any final comments. Given that previous policy is based upon the fallacy of de-identified information, how difficult will it be to introduce new policies that allow control by patients?
This is Debra. I think it will be difficult. I think it will be gradual. But there are, I mean, just in the last year or two have been huge strides in this direction. And I think that once researchers see the benefits to them, that is, you know, the more people trust you, the more willing they will be to participate in research. But of course, you have to earn that trust. And if this, that is ceding some control to participants in your research, actually increases trust and increases participation, then the scientists will come along.
Okay, excellent. Any other thoughts from Yaniv or Robert?
I agree with Debra, but I also think it's gradual because the incentive systems, the mentality, I think, and some of the regulations have actually sort of guided people to siloing information and to taking the opportunities provided by loopholes in the privacy laws that are created through exclusions and through capabilities that were under the old regime. There's a resistance to moving and to ceding the control that Debra talked about. It's a natural human characteristic, but I totally agree with her assessment that as researchers and as clinicians and providers, health groups, payers, and governments start to see the benefit of ceding the control to some degree back to the individual, that there's reason for hope.
Great. And then any final thoughts, Yaniv?
I pretty much agree. I think you read the newspapers and you see the poor state of privacy in general. So the problem is not just about genetic information or medical information. We see that in other aspects of our life. So definitely should be some more thinking from many stakeholders in society about what we want. How is the future society of and what is the role of personal information in that?
Excellent. Well, we are officially out of time. I do want to thank our presenters today. You were fantastic. As a reminder, this webinar will be archived, both the slides and the recording of the session itself. And please sign up for our next deep dive, our privacy requirements in whole genome sequencing, different in various contexts, life course differences, severity of disease, and non-medical uses. And once again, that session is on November 12th from noon to 1 p.m. Eastern Time. Thank you very much for joining us today. And if you have any other questions, feel free to email us at Genetic Alliance, and we'll pass them on to the presenters.